Welcome back, everybody. I hope you took a small break there and that you're ready to go, because we are going to be continuing on with this conversation of the nervous system. In particular, we're going to be focusing on the big overarching parts of our nervous system. As you probably recall already, especially if you transitioned quickly over to these slides, in the first part of this class, we talked extensively about the neuron, its structure, how it communicates with each other, and we said that understanding the neuron was critical because it's the building block, the main component of how our nervous system actually works. But as neuroscientists, we know that as we move beyond the neuron into the structures and the connections, we see another story that needs to be told, more information that needs to be studied, so we can truly understand how we're capable of doing all of the different activities that we did in the beginning of this particular class. When we talk about the nervous system, what we usually do is we break it down into two major parts. The first major part we have is something called our central nervous system, which involves what's called the cerebral cortex, what most people often call the brain. The inner portions of that cerebral cortex, we have the midbrain, the hindbrain, the cerebellum. We also have the spinal cord. This is where a huge percentage of our neurons within our nervous system are actually located. But in addition to having the central nervous system, we also have something called the peripheral nervous system, or PNS. And this is the collection of nerves, neurons all over our body, that are allowing our body to communicate with our brain and vice versa. When we look at the amount of neurons that you have, most people again have roughly 80 to 100 billion neurons in their body when they're adults. And we know much more about this nervous system than just the fact that you have that many cells. Instead, what we've started to recognize over the years is that cerebral cortex in particular, and the rest of our body, I guess, when we're talking about the nervous system, is arranged in very specific ways that are almost universal for everybody. And this arrangement that we see within our body is primarily focused, in the mind of a neuroscientist, on that cerebral cortex. Because when we look at the human brain, in particular we look at a person who's active and using those neurons, what we see is that the human brain is arranged in a way where specific regions of the cerebral cortex and connecting areas are always associated with specific types of tasks. Now, one caveat to this brief piece of information before we get into breaking it down is the fact that even though in pictures you might see a pretty brain like the one you see up there, if we take any of your brains and put it under a microscope, each person's brain, or even just take images of it, looks very, very different. In fact, your structure is probably nowhere near the structure of the brain that you see there in the picture. Instead, each of us has extra crevices, tiny movements, parts that didn't grow quite as well as other ones, but that doesn't mean that your cognitive functioning is somehow inhibited because your brain doesn't have this nice symmetrical shape to it. Instead, it's just a natural part of the growth process. But when we look closer at brains, when we try to kind of remove some of the small individual differences, what we're often surprised to find is that your cerebral cortex is very similar to the cerebral cortex of almost everybody around you. One of the first major things that we've recognized over the years is that the cerebral cortex is actually separated into 4 different things that we call lobes. You've got the frontal lobe, which is in the very front of your brain, on both the left and right side, which controls things like higher order processing, controlling ideas, planning. You've also got areas right behind it called the parietal lobe and temporal lobe that are there for very specific purposes. The parietal lobe is heavily involved in our sensory and our motor systems. And what's really interesting with the parietal lobe is the fact that the connection between the brain and our body is somewhat unexpected. In essence, what we see when we maybe move our left hand or try to raise our right arm is that the side of the brain that controls those functions is actually what we call contralateral to the body that we see. In essence, if you raise your left hand, it's actually the right side of your brain that is allowing you to do this. If I touch your right foot and you sense it, It's the left side of your parietal lobe that actually makes sense of that contact, that processes that particular piece of information. And that is something that's really surprising to psychologists, but we think there might be some evolutionary reason as to why that developed. However, for most people, contralateral communication, whether or not we know for sure why it's there, does exist. There's one exception to this, and that's the fact that a large percentage of left-handed individuals do seem to actually not have contralateral communication. And some people who are left-handed actually have bicommunication. So both sides of their brain are communicating with one particular side of their body. But for most of us, even left-handers included, contralateral communication actually happens. I guess technically there are a small percentage of right-handed people that have that exception, but very, very few of them do. Another lobe that we have, again, in addition to this parietal lobe, is our temporal lobe. This is, again, something that's very hemisphere specific. This particular lobe is involved in auditory ability, but it's also involved in a lot of very complex behaviors that we exhibit on a regular basis. One of the main things the temporal lobe is associated with is language. So our ability to process what's being said, speak, form a sentence, all of these things are done, at least for the most part, inside the temporal lobe. What's interesting to a lot of psychologists is the fact that even though language is something that involves hearing very often, only one half of the brain is usually involved in the production of language. It's on the left side of the brain that we actually have this ability to produce language. That particular side is also associated with scientific and math skills, logic problems, and a number of analytic abilities as well. If we flip the brain over and look at the temporal lobe on the other side, instead of seeing a huge amount of that particular section of the brain dedicated to language, instead we see thought, intuition, and creativity being the dominant parts of this particular temporal lobe. Again, for left-handers, this sometimes flips, but for the most part, a large percentage of the population has these allocation of resources in the temporal lobe. The last major lobe that we have, one that's exceptionally important for most of us, is something called the occipital lobe. And this is the area that's also sometimes called the visual cortex because, as you might guess, it's highly involved in the process of vision. And if you remember back to the idea of slices and Kajal's work and Camillo Golgi's work, and you probably remember us talking about clusters of neurons that were situated along that occipital lobe. Well, that's because as we go from the back of the occipital lobe towards the front, you're actually doing higher levels of processing as you go. Basic processing, like picking up on where an object was, what color it is done in the very beginning, or what's called the V1 cortex of our occipital lobe. But when we start to determine what we're looking at, where it's going, how to interact with it, we move up to higher levels of our occipital lobe as we move up into our parietal lobe for possible movement, temporal lobe for interaction, and even frontal lobe to actually coordinate plans as to how to interact with what we're experiencing in our environment. All of these parts interact with each other tremendously. What you see if you get into a higher level class is actually, even though we break it down into very simpler forms, it's much more complex than what we have. But for basic levels here in an intro to psych class, I need you to know that hemispheres matter, that lobes matter, and we can look at the brain in very complex ways if we get into the scientific analysis of all the different things that our brains are capable of doing. Another thing that's interesting to a lot of psychologists is that if we go below the cerebral cortex, we see other things that often are called systems. Small, tiny structures embedded below the cerebral cortex that are involved in other specialized types of functioning. The picture on the left is a picture of what we call our auditory system, where we start from something called the cochlea, a structure right behind our ears that has tiny hair cell follicles that allow us to actually make sense of the vibrations in the air. And those cells that go from the cochlea actually travel into areas within the brain, like the ventral collicular nucleus, the lemniscal nucleus, and the medial geniculate nucleus, so we can eventually make sense of all the sounds that we have and also where those sounds are coming from. That means by the time we get to our cerebral cortex, a large amount of information about what's going on has already been determined or figured out by the lower regions of our brain. This happens for a number of different types of systems in our brain. Another great example of this is what we call our limbic system. And this particular limbic system has a number of parts to it, but some of the more prominent parts that we have in this particular system is our olfactory bulb, our hippocampus, and our amygdala. And that's because these three specific regions are involved in very important functioning in the human body. Our olfactory valve is associated with, if you know the word olfaction, you probably know already, but it's associated with smell, our ability to process the different scents that are in our environment. And in fact, we have neurons dedicated to differentiating between different types of sounds. What's really interesting to a lot of psychologists is that even though we know this area is involved in processing smell, we don't quite know how it actually determines the differences between scents in our environment. This olfactory bulb, though, as I said, is connected to the limbic system for a very important reason. One of the major reasons why it's connected to this system is because of its link to the hippocampus, which is the region of the brain that we strongly link to memories, our ability to have both short and long-term memories. This connection between the olfactory bulb and the hippocampus allows us to form memories very easily when scents are actually presented to us. Example of this for any of you could be walking around somebody that you've never met before who happens to be wearing a perfume or cologne of a loved one or somebody that you're well acquainted with. What often will probably happen is that smell will quickly bring up images in your head or your mind of that individual that wears the same perfume or cologne. In fact, walking into a room with roses, if you've had a past experience with those, could possibly bring up other memories. And that quick automatic response of memories to the olfaction is because of the fact that this limbic system has all of these parts really integrated with each other. That third region that we said is really important in this limbic system is the amygdala, which is highly involved in emotional processing. So when you're happy, sad, or anywhere in between, or experiencing any of the emotions that really don't fit quite on that spectrum, it's because of activation in this amygdala. Again, it's closely linked to not only the olfactory bulb, but the hippocampus, which means our smells can produce very strong emotional reactions. Anybody who's walked into a room that somebody's left, unfortunately, with a very unpleasant odor, you know the emotional experience that you have after that. Or if you've ever walked into something that's very pleasant, you also know that you can have a positive emotional experience as well. Same thing happens with memory. In fact, most researchers now believe that one of the reasons why some memories are much easier to access than others is because of the emotional connection that we have to them. So when we look at the limbic system, our auditory system, I want you to appreciate how they work, but also understand that they're just two of many, many systems that we have within our brain. As neuroscientists, we're always trying to break these down and understand them at much more complex levels as we progress in our understanding of the system. One thing you might be asking yourself now, though, is how do we know these things? If we've only been looking at the brain for 20, 30 years, there has to be some way of actually proving the information that we're actually studying. Well, instead of spending the rest of this class talking about hundreds of systems or different nuances to the brain, what I thought we'd do is actually explore what neuroscientists are doing nowadays in order to understand how the brain actually works. When we look at this process, what we see is that it's not just one thing that scientists are doing. Instead, we have a plethora of different approaches to understanding how the human brain and our nervous system actually works. One of the first ways we started exploring the brain and the nervous system was not actually by using humans, but instead using other animals that we thought had similar systems and structures to human beings. More often than not, in our early years of experimentation, especially even in the behavioral world, we used rats and mice. Nowadays, we've gone up to looking at pigs, and primarily we've also started looking at primates as our kind of analogous animal to the human brain. What we'll often do is not only look at structures, but try to find ways to perfect our understanding of how the brain works and communicates with its body. There are major ethical concerns to using animals nowadays, and in fact, a large number of neuroscientists, because some of the other tools we've come up with, which we'll discuss later, have abandoned doing animal research altogether. But there are still places where animal research has still been used, and it can be used for very important purposes. The image that you see in front of you is quite possibly one of those very important purposes that are out there. What you see in that picture is a monkey that's actually connected to a computer. If you look at his head, you'll notice a portion of his skull is covered by a metal cylinder. Well, underneath that metal cylinder, what you actually have is the monkey's scalp removed, his skull opened, and a number of measurement tools hooked up to a very specific region of his parietal lobe, the area that's involved in movement. Those connections are sent to a computer, which are then sent to that electronic arm that you see in front of you. So if you look in the upper left-hand picture, what you'll see is a monkey actually moving that electronic arm with its brain activity. In front of the monkey at that point is a sugar cube. Picture to the right of it, the monkey's now closed the hand to grab that sugar cube. Then he's been able to move the arm back to his mouth, and after placing the sugar cube into his mouth, he's able to release the fingers on that hand and pull the hand away as he's chewing on the cube. This behavior, this task, is really amazing to many neuroscientists, because what it tells us is that someday we might be able to create machines, computers, that would replicate the movement abilities that we have with our brain being connected to our body if something were to, even though we would never want it to happen, actually happen to our body. Say you lose a limb in an accident or in a war. What could possibly happen from this research is we might develop the tools to replace that limb, give a person the ability to move that arm that no longer exists in some way that's resembling what they were capable of doing originally. And all that would be needed was just a thought, the same thing that we see nowadays in most of our behaviors. I don't want to convince you that animal research is a good idea. In fact, there are many, many faculty members and people in the area of psychology that say we should do away with it entirely. But if there is an argument to be made for continuing animal research, this very well might be one of the cases where studying this, understanding things, and perfecting it so we could possibly make many, many individuals' lives better, it gives this particular type of research some credibility. Another way we study the brain, when we look at kind of damage or ways to kind of break it down, by not instead of studying animals, but instead looking at human brains who have gone through some type of traumatic experience. A classic example of traumatic experience research that's been formed over the years is research on two particular areas of the brain that we associate with language. At the time, two particular researchers looking at understanding language were Paul Broca and Carl Wernicke. Both of them thought in their studies, which were actually presented at almost simultaneously during different conferences, that they had discovered the source to language. In fact, both Paul Broca and Carl Wernicke had a number of patients that had suffered from damage to a very specific region of their brain through strokes or other types of damage that have occurred over the years that somehow let these people still live. What Wernicke and Broca found in their studies was that by having damage to this region, these people were almost incapable of communicating. But what's interesting is the fact that these areas were very different from each other. And the effects on language were very different from each other. In Wernicke's patients, the one you see on the right, people looked like they understood language. Somebody would talk to them, they would sort of make sense of it, and then they would produce a stream of consciousness that had nothing to do with the question being asked. And oftentimes the words were completely jarbled, and it just sounded like what we often call in psychology word salad, a mixing of random sounds and words put together in something that for some reason, because of the timbre, the structure of the voice, sounds like an actual conversation. What's really interesting with Wernicke's patients is the fact that even though they're speaking gibberish, they have no idea that they're not making sense. And in fact, most of them only get a clue that they're not making sense by looking at the cues that the listener is actually giving them. Broca's patients, however, even though they also spoke gibberish, do show very, very pronounced frustration when they're trying to speak. In essence, it's as if they formed a sentence, are ready to produce it, and somewhere between coming up with the sentence and actually speaking it, something gets lost. So for Broca's patients, even though their language deficits are almost identical to Wernicke's patients, the frustration is noticeable, the anger is noticeable, and oftentimes they kind of dance around things until they find a way to produce something that gets their ideas out. And when we look at cases like Broca's patients and Wernicke's patients, we do have the ability to understand some of the major portions of ability that are controlled. Sorry, we're back. This particular type of research is being done today with people who've suffered from strokes or have had other types of damage. But, as you might guess, there are some major problems with this type of research. These problems often accompany the benefits that we have with research on animals and on human subjects with damage to their brain. One benefit we do have with this research is the fact that we do have the ability to determine what we call necessity of a region for different processes. So if I have a region of my brain that's gone and suddenly I can't do something, I can know with fairly good certainty that that particular area probably had some major role in our functioning when we were doing things. It doesn't necessarily mean that we're not using other parts of our brain for different types of functioning. When we look at weaknesses, though, one of the big things we see is the fact that when we use animals and when we use individuals with damage to their brain, we have a huge lack of control. Namely, if somebody suffers a stroke, we can't tell them to have a stroke in only a specific area of their brain. If somebody experiences damage through, say, a rod shooting through their head, like in the case of Phineas Gage, which you'll read about in your textbook, we have no way of saying only have that rod shoot through one specific part of your brain. When we look at animals, we can't be 100% sure that an animal's brain is perfectly analogous to a human's brain. So instead, what we have to do is just hope and guess that the damage is only located in specific regions and find enough people that overlap for us to have fairly good certainty that one specific region is indeed the important part of a specific type of functioning. But another problem we have with this is that even if we know that a specific region is associated with something, it's really tough to fine-tune exactly which types of functioning are associated with that region. And that's because as people start to heal, as they start to recover from their damage, the brain has an uncanny ability to almost what we would call rewrite itself. What we see is that those neuron clusters that were dedicated to original functions can actually be replaced by other neurons, which were originally set out to do other types of tasks, in order to allow us to do things that we temporarily could not do. A
Classic case of this would be people with actual Broca's aphasia. And we see with a lot of them is that even though they lose the ability to speak for quite a substantial period of time, numbers of them find ways to communicate, maybe not at the same level, but at least at a rudimentary level that involves language, spoken language. And that's because their brain will actually repath itself, go around the damaged area, and create new structures that allow the human body to kind of be able to do the things that it was doing before. This ability is something we call plasticity. And when we look at young children, we see high amounts of neuroplasticity. In fact, there's children that could have entire regions of their brain not functioning or die off and still function as if they're completely normal. As we age, neuroplasticity lessens, but there's a number of studies that have shown that we do have some neuroplasticity even in our late years. A third major weakness of these types of studies is the fact that there is not many people that actually survive damage to their brain. And for ethical purposes, we don't want to test on hundreds of animals to try to figure out different regions of the brain and what they do. So no matter who we look at, we don't have enough information unless we have large samples. And large samples just don't happen easily. If you have damage to your brain, odds are you're probably going to die. So very few people are going to survive these types of damages and allow us to study them. And honestly, people have lives. We don't want to invade them too much if they've had this type of damage. So even if we have access to some people, we don't have a ton of access to them 24 hours a day. And that means we'll look for other ways to try to understand the brain.
One of the ways we've started to almost replicate these lesion studies is through a new type of experimental research called TMS studies, or transcranial magnetic stimulation. It's a slightly sarcastic picture to the right of what TMS looks like, but you can get a sense of how this process works by looking at that picture. Because what we do in TMS studies is essentially replicate lesion studies in healthy individuals. And the way we do this is in the name. What we do is we actually send magnetic pulses to very specific regions of a person's brain. And by sending these pulses for an extended period of time to that brain, we can actually temporarily deaden that portion, essentially turn the neurons off for a short period of time, and then test the functioning that a person loses while that area is deadened. The rule of thumb with TMS is for every minute of magnetic stimulation, where the pulses are hitting a region of brain, you're going to knock out that region of the brain for a minute. So if I want to see a part of the occipital lobe, that region again involved in vision, for a specific type of vision, I can send a pulse to that region for say 20 minutes, and I have 20 minutes then to test a person and see which type of functioning they've lost. The TMS is a relatively new type of research, so we don't have a ton of information on it, even though there seems to be no evidence to show that it's dangerous to people and that functioning is completely regained. The only long-term effects that we've seen is the fact that people tend to have slightly more elated moods for a number of hours after they've experienced TMS. But most people think that's probably just a side effect of being in part of something really interesting and novel. However, when we look at temporary offsets or problems with TMS, there are some more obvious ones. That picture to your right shows a person grimacing or acting weird primarily because it's kind of an annoying process. In fact, your instructor went through a number of TMS studies when he was a graduate student, helping the two gentlemen that you see on your right with some of their experiments. And most of the time when you're in TMS, what you'll have is those magnetic pulses not only deadening a region of your brain, but also passing through a number of nerves that you have on your cranial nerve. And anybody who's had action or activity there knows that having those nerves activated can be somewhat annoying. It's very painful, and it also causes a number of twitches that you have in your face. So each time you get pulsed by the TMS studies, your face twitches with each individual pulse. So imagine sitting there for 20 minutes
with your eye constantly twitching with each pulse as you try to deaden a region of your brain and get rushed over to be tested on something, only to go back and have that region of your brain get deadened for another 20 minutes. It's somewhat annoying, but it's a great, great tool that we have at our disposal nowadays because it allows us to do those lesion studies without having just random chance determine what gets knocked out. And with being able to use healthy, normal individuals, it will show no long-term residual effects. It also allows us to get around that plasticity idea that we talked about earlier. But even with TMS, lots of researchers have said maybe we need to stop looking at necessity and instead just look at what's actually being used in our brain and adjacent areas when we're doing specific types of tasks. And that's led us into some very different types of research that don't involve the ones we've discussed so far.